Hi, my name is Rodolfo Barangu, and I'm the editor-in-chief of the CRISPR Journal. We are delighted to be sponsoring today's special GENLive episode featuring the great Walter Isaacson discussing his new book, The Codebreaker. The CRISPR Journal is one of over 100 peer-reviewed journals published by Media Libre Inc., the publisher of Gen. We launched in 2018 as the journal by the CRISPR community for the CRISPR community. We publish original research on all aspects of CRISPR biology, evolution, genome editing, and applications thereof. And I am proud to lead an exceptionally diverse editorial board that includes our very own Jennifer Doudna and others of other CRISPR pioneers around the world. We're also a forum for discussions of ethical and regulatory issues around the practical uses and even the misuses of genome editing. 2020 was a great year for us. We received our first impact factor. The submissions doubled, and we published a number of very interesting articles I suggest that you read. We hope you will consider us for your next paper, and you can find out more at CRISPRjournal.com. Enjoy the show and read this fantastic book.
Hello, everyone, and welcome to a very special edition of GENLive, our monthly hangouts with the editors of Gen, Genetic Engineering and Biotechnology News. We have a very special guest today joining us from his home in New Orleans. I hope I said that right. The one and only Walter Isaacson, the author of a great new book, and we'll be getting to Walter in just a moment. Thanks to Rodolphe Barangu, the chief editor of the CRISPR Journal, for that wonderful introduction. And let's just quickly meet the folks at Gen. I'm your co-host, Kevin Davis. I'm an editor with Gen and the CRISPR Journal, and the author of a recent book strategically positioned over my shoulder, Editing Humanity, the CRISPR Revolution and the New Era of Genome Editing, which I was absolutely thrilled to see cited in Walter's book a couple of times. I'm joined, as usual, by a clutch of Gen editors, John Sterling, the chief editor of Gen, Juliana Lemieux, Dr. Juliana Lemieux, senior science editor, and Alex Philippidis, senior business editor, holding the fort in the Gen newsroom there in New Rochelle, New York. Hi, everyone.
Hey.
Howdy.
Kevin.
We also welcome your questions from the audience at any time over the next hour for Walter. So please use the Q&A box, and we will try to squeeze in as many of those as we can in the final portion of the program. The story of CRISPR has, of course, captivated scientists and the public alike, spawning countless magazine cover stories, books, documentaries, films, new journals, of course, a biotech revolution, and the 2020 Nobel Prize in Chemistry. But one of the biggest accolades that this field, or indeed any field, can receive is to be profiled in a new book from the best-selling author, Walter Isaacson. His new book, as I'm sure you know by now, is The Codebreaker, Jennifer Doudna, Gene Editing, and the Future of the Human Race. Doudna's story, Walter writes, is a thrilling detective tale that involves the world's most profound mysteries, from the origins of life to the future of our species. Walter wears many hats. He's a professor of history at Tulane University. He's a former CEO of the Aspen Institute, former editor of Time magazine. He's a regular host on Amanpour.co on PBS, and the author of multiple best-selling books that many of you have read, including Leonardo da Vinci, Steve Jobs, and Einstein, His Life and Universes. The Codebreaker was published last week by Simon & Schuster, and Walter has barely been off the television since. Walter, a very warm welcome to you to GENLive, and happy St. Patrick's Day. I imagine it's something you take pretty seriously down there.
Well, I certainly, in the back of me is the balcony overlooking Royal Street. And unfortunately, everybody seems to be drinking very green drinks and singing. So I've got the shutters closed.
Well, I hope I know you're giving a lecture later on this afternoon. So hopefully you can go join them at some point. It's socially distanced and responsibly, of course.
Thank you. Let me just start by thanking you. As you say, you're not only cited in the book, but I had to redo some of the citations because I cited you in the CRISPR Journal, I think, and Rodolphe as well. The CRISPR Journal had so many good interviews with the main characters in your book, Editing Humanity, and in mine. And so I cited them all from the magazine, but then also went back, read the book, and found more things in it. So I threw them all in. So thank you for your great work. As I showed you, I'll put it above my shoulder too, because it's an invaluable work about all the issues, especially when you deal with He and the reaction to the Chinese twin babies.
Well, we're not here to talk about that. We're here to talk about the code breaker. So let's. What's the last week been like for you? You've been on a whirlwind. I know you've not hardly left your chair there, but it's been a whirlwind virtual tour of sorts.
Yeah, usually when you do book tours, you go to Kansas City and you go to a few places. Here, it's in the waning days of the COVID pandemic, or what I hope are waning days. You just sit here and you're on TV. And unfortunately, people are sitting at home watching TV, so they've heard you say the same thing 8 or 9 times. I'm going to try really hard to actually contradict everything I've said in all previous interviews.
So what was the?
By the way, when Rodolphe is one of my heroes. That was sweet of you to have him do it. But I just want people to show, among other things, I got Simon used to do was color pictures. But that's Rodolphe without the goatee, looking very young, looking very, very good looking. So he's the hero of one of the yogurt makers is one of the chapters.
Oh God, his head is just going like crazy. What was the genesis of the book, Walter? Because you're not known for writing about the life sciences. So did you have an itch to turn to the life sciences after all these other great books you've written, or was it inspired by Jennifer? How did the book get off the ground? I
kind of realized that there have been 3 great innovation revolutions of our time. The physics revolution in the first half of the 20th century, coming out of Einstein's miracle year papers, where you get everything from, the atom bomb to semiconductors to space travel. Likewise, after we figure out the atom, we do the bit, another great kernel of our existence with binary digits, and you have the microchip, the computer, the network. And I did that through Steve Jobs. I think the most consequential revolution will be the life sciences revolution. So about 6 or 7 years ago, I started saying, what's a good thread to get me into that revolution? I'd always been interested in it. And I ran into Jennifer when she came to Aspen, and we did some events together. And she told me the story of her dad giving her, Jim Watson's, a double helix in 6th grade. And I went back to my home. Actually, my dad was still alive then. My home in New Orleans I grew up in, and I found that old copy of the double helix. It was beautiful. It had that pale red cover. And I realized I'd gotten it in 6th grade. It was sort of inscribed. And I'd made all sorts of marginal notes of words I didn't understand, like biochemistry, and trying to show what that is as a 6th grader. And I realized how beautiful the life sciences are. And what people like you do, or, Rodolphe does, or, people in the life sciences, even before this pandemic struck, I thought, boy, this field needs more publicity. And frankly, the digital revolution maybe needs a little less publicity. We've had enough profiles of, Mark Zuckerberg or Elon Musk, whatever. And so I just thought this was a field that was incredibly important, but underappreciated by the average person who would not, at least until she won the Nobel Prize, know the name Jennifer Doudna, Rodolphe Barangu, or, Francisco Mojica, or all the people in your book and mine. And so I thought, and being sort of having a biographer's instinct, I liked finding a central character. And for Jennifer, it starts with the double helix and it ends with fighting COVID and doing the ethical issues. So she's a good central character to follow.
Yeah, there's a lovely scene in the book where you say, I'm a proud owner, you are a proud owner of the first edition of the double helix. And it'd be worth something, except you'd handwritten, penciled notes in every margin. So
yeah, I can't sell it on eBay. It was a first edition.
Can't. Yeah, you dedicated the Codebreaker to 2 women who recently passed away, but one of whom was Alice Mayhew, your longtime editor, who edited the Horace Phelan Judson classic, The Eighth Day of Creation. So it's a wonderful lineage here.
I'm amazed by you, Kevin. Not only have you read the footnotes, you've read the acknowledgments. I think you've read this book better than I have. Alice was truly wonderful. And, I thought, okay, I've done Leonardo. Usually I go to lunch with Alice Mayhew. She's been around forever. She's no longer with us a lot. And I really just say 2 or 3 words, and we figured out. I'll go Leonardo, or I'll go Steve Jobs or Einstein. And this time I had to say, okay, I want to do a very little known or unknown biochemist with a slightly hard to pronounce last name who helped invent a technology with a very cute acronym that nobody can remember what it stands for.
Yeah.
And I want to do biotechnology. And I thought she would say, look, you're out of your mind. Why don't you do Aristotle? Why don't you do whatever? And she said, oh, I love it. A Journey of Adventure. My favorite book I ever edited was Horace Freeland Judson's, Eighth Day of Creation.
Yeah.
Something I had read a long time ago, but then picked up again.
Yeah.
And I was just blown away by how the late, great Alice Mayhew, every time I'd raise something, she'd say, oh yeah, don't you love the way Judson went to the Cavendish lab? And when he was there, he describes this. She knew from memory every sentence in that book. That book is a great book. And with all due respect to you and me, if people need to start with a book in this field, they should start with the Eighth Day of Creation.
But it's funny, you said something that I think we'll follow up. You threw yourself into the community. You went to the conferences, you hung out in the labs, you did CRISPR experiments. We'll ask you a bit more about that later on. I have one more question and then we'll pass the microphone, the baton over to John. So you mentioned meeting Jennifer at the Aspen Institute. Is that actually in Aspen or is that in DC?
The headquarters is in DC, but the campus is in Aspen, and that's where we met, was in Colorado.
But there was something, and then you presented this idea sometime later to your editor. What was it about Jennifer that you saw she will be the vessel through which I will tell this story?
Well, to be honest, I didn't. It wasn't like one day I just decided that. I thought, okay, this is a good narrative thread. It has to start somewhere. It should start with somebody picking up the double helix. As it went along and as more and more policy issues happened, as Ho-Joon-Key happened, while I was working on the book, and then as the international summits, and then the pivot to COVID, she seemed to be involved in each of those. And so initially I once wrote a book called The Innovators, which was a group book, a little bit like Editing Humanity, but about innovation in the digital age, which wove it all together. But this time I wanted a central character. And the more I played with it, and I'd go to MIT and I'd hang with Eric Lander and he would offer alternative ideas, let me say. I loved meeting with Feng Zhang. He's just, as you know, and I'm not sure Jennifer Doudna fully knows, is one of the nicest, most genuine people. And I wanted all of them to be in the book, but as it went along, Jennifer became the central character, I'll say. It's not really just a biography of her. It's a joint biography with one central character.
Great. Let's have your questions for Walter Isaacson. Please use the Q&A box over the next 30 minutes or so. John, over to you.
Yeah, well, by the way, we live in a very insular world covering the science universe. And the fact that your book, I thought, not only doesn't compromise the science, but it's readable. I think it's great because we're getting this news out to everybody who's interested. I've been covering this field 35 years, and I remember when people said biotechnology, I'd see a little slight fear and tension in their face. So it's different. And in that vein, covering the biotech for all these years, every one of these technologies, immunotherapy, synthetic biology, AI, one time you could do some science fiction, and now they're here. What was it about CRISPR, in addition to Jennifer, that really caught your attention and said, not just Jennifer, but this technology is really something?
Right. Well, I think the Nobel Committee said it well, as it brings science into a whole new epoch, and it really connects with the humanities. All of the people I've written about, from Leonardo to Steve Jobs, are people with one foot in the humanities and one foot in the sciences. And although, recombinant DNA and the founding of Genentech, all of which is in the book, all very important, the notion that we can edit the human genes is, I think, a game changer for a species. I mean, we're the first one to come along that says, all right, we can just edit our DNA. It's also something people can both relate to, be sometimes taken aback at the concept, but then have to process. So it was a good way into, I hope, a lot of other subjects in the book that aren't just gene editing. But as Jennifer found out as a young, a kid, the structure of molecules is actually quite interesting. And nowadays, knowing the structure of molecules, as Rosalind Franklin did, as Jennifer did, and others in the book, we can recode them. And so that just seemed a better technology to look at than, let's say, immunotherapy or something, which I think is also incredibly important and deserves its own book.
Mm-hmm. And in light of the three people you mentioned, Jobs, Einstein, and Leonardo, in your book, you point out that they were kind of outsiders and they never quite fit into their society. And yet, look what they did. Yeah. Jennifer, some similar aspects there. How does she fit into that paradigm?
Well, she told me over and over again that she felt so much like an outsider when she was growing up in Hawaii, alienated, depressed, insecure. I mean, she was very open about it because she was a tall, lanky, blonde girl in Ilo, Hawaii, where the rest of the students were Polynesian descent. And they made fun of her. They called her haole, which isn't quite as bad as it sounds, but it sounded bad to her when she was a kid. And I think people who are outsiders often think different a bit. And when she said, it made me, when we played soccer, not run towards the ball and not play the positions everybody else wanted to play. And she said, and that's probably why I ran towards RNA when everybody else was running towards DNA. But whether it's Leonardo or Steve Jobs or Einstein, you sort of say, wait, how do I fit into this cosmos in which I somehow find myself? And I'm not sure how I fit in. So let me find clues about how we all fit into this cosmos. So I found that it wasn't something I would have noticed had she not talked about it a lot and said that spurred her curiosity.
Yeah, it's common in science. Kevin, we have a lot of questions. I want to turn it over to one of our colleagues.
Sure, let's go on to Juliana, please.
Sure, yeah, thanks. So in writing this book, you went to scientific conferences, spent time in labs, even did experiments, and talked to many, many different scientists. So I'm wondering, did you find anything unique about scientists or the scientific culture?
Well, yes, and it's particularly, unfortunately, if not unique, rather rare in this day and age, is you'd be at the bench and somebody would have this incredibly good theory about some way, a kidney cell, human kidney cell might be cut and what would happen. And they'd do the experiment and it would turn out not to be that way, and they'd revise their theory. When I wrote about Ben Franklin, what made him the great founder in many ways is that unlike John and Adams and his cousin Samuel or Jefferson, he didn't have a whole lot of fixed ideas. He kept saying, let the experiment be made, whether lightning was a spark of electricity or whether we should have estate taxes or welfare laws. And so that scientific method, that's sort of the hero of this book as well for the lay reader, which is in this day and age where people are anti-science, some of them, dubious about everything from climate change to vaccines. I want to demystify science. And part of it, something John said that resonated. Every now and then, people who are in the priesthood of great scientists will say, well, you're just a popularizer. And I tend to say, well, thank you. Because I do think there's a role for the person who tries to make it accessible to the lay reader, because if people are afraid of science, they're not going to fund it. They're not going to follow it. And I think sometimes, science has become frightening to people. And that's another reason to do it through a Jennifer Doudna, or I could have done it through a Fong Zhang, but through somebody who at the beginning of this book has to go pick up her 17-year-old boy from robot camp because she's afraid the pandemic is starting to spread a year ago. And so it tells the ordinary reader who, instead of grimacing, as John said, when you use a word like biotechnology, it tries to bring the ordinary reader into a journey of discovery.
Yeah, extremely well said. I couldn't agree more myself, especially at this moment in time. And as a scientist, it felt very familiar reading the book, the descriptions. So thank you for that. So a theme in your book is the collaborative nature of current day science. But many scientists oppose the individual nature of the more traditional awards. So how do you feel about Doudna and Charpentier winning the Nobel Prize after or during writing a book that highlights the contributions of such a huge cast of characters?
Well, I'm going to be perfectly honest with you, which is unlike Jennifer. I set my alarm at 4 a.m. the day I knew a prize would be announced in chemistry. She slept through the phone calls from Stockholm, as Kevin knows. But I'm here in New Orleans. It was 4 a.m. and they're live streaming from Sweden. And when they say for rewriting the code of life and bringing science into a new epoch, I knew it'd be CRISPR, and then held my breath to see who the three I thought would be. So I don't want to be disingenuous and say, I think, I'm sorry about the Nobel Prize. But I'll say something which probably is going to sound bad if it gets spread too much. I think science prizes, on balance, probably nowadays do as much harm as good. They do, as you say, try to single out 1, 2, or 3 people and they tend to disincent not only collaboration and teamwork, but also generosity in saying who deserves credit for things. In fact, there are 2 things that I think are disincentives: patents and prizes. And they both exist for really good reasons. Like Facebook, I think it did more good than harm usually. But after a while, like Facebook, I think some things actually become counterproductive. And when COVID hit, everybody started becoming much more collaborative, sharing things more easily, not asserting intellectual property rights. When I talked to Fong Zhang, I don't want to go into it, I guess, because it wasn't on the record, but it was after the Nobel Prize. I won't quote him because it was just a private conversation. But among the things I sense from him is, well, in some ways, it's now a relief. The prizes are done with, so to speak, from the Kavli to the Nobel to the breakthroughs or whatever. And now we can all get back to praising one another and not worrying about them. So I kind of felt a sense of relief too once the Nobel was done. It certainly helped I keep showing the color pictures, but I had to really push Simon and Schuster to do it at no extra cost with the color pictures. But, there's a picture of Jennifer in her kitchen.
Kitchen, right?
Yeah, with Jamie and Andy. And it's just a good way to sort of culminate the book is, when she gets to go get a cup of coffee and the TV crews have already shown up and somebody takes a picture. So it was a relief. I didn't have to worry. And I think it may be a relief to all scientists in the CRISPR field that they don't have to jockey quite as much. I actually hope, I'm sorry, I'm going on a bit. I'll say something for this very technically oriented crowd too. When Eric Lander tweeted it out and said, praise Jennifer and congratulate her and Emmanuel, and said that he was also excited about the advances that were coming for patients in this field, it made me think that the Nobel for Medicine which is not, they don't usually award it to the same general area in two fields. I do think that Feng Zhang, George Church, and David Liu, but I'll ask you all to nominate others, should be up for the Nobel in medicine once we get it truly into out of clinical trials and into the bedside.
Yeah, it's a good trio, and I'm thrilled that you made that point. I definitely think the basic fundamental technology of CRISPR's received the ultimate accolade, but the therapeutic applications are still out there. It's still a long way to go there. We have to stress that. Julianne, I think you probably have one more question, and then we'll move on to Alex.
Actually, my last question will be a quick one, which is the code breaker. I can guess what that title means, but can you tell us how did that come to be?
Well, I was interested in digital code, and it's sort of supposed to take us from the digital revolution to the genetic and life sciences revolution. And when Jennifer Doudna is able to do the structure of RNA and show how that structure leads to so many clues about how life works, and when she and Emmanuel Charpentier and others figured out how you can recode the single guide RNA that takes the Cas9 enzyme in a CRISPR system to edit. And then when the mRNA scientists showed how you could recode mRNA for each new variant, I realized we were entering a new era of coding. And having written about Alan Turing and Ada Lovelace, who were code breakers, and having written about people in the digital revolution like Steve Jobs, I felt code breaker was a term of endearment, at least for me. If you'll hold up, Juliana, the book again.
Sure.
And just so I will give a small little insider's note. It's hard to see on that cover, but around Jennifer as the code, you can see, I think, GACCGBG, whatever. You can see letters and a halo around her. That is a snippet of RNA code. And you see the letters?
Oh, yes. Now I see it.
And they're around, yeah.
Yeah.
And those letters are the code for the part of the fragment of the spike protein that bonds to a human cell. I took it from the sequence that Pfizer used just as an insider knowledge. Okay, we broke that code as well.
A little Easter egg there. We have a question, Walter, from a fellow author, Victor McElhaney. It's great to hear from you, Victor. Can you say a word about Jennifer as an expositor, a popular teacher, a person aware of ethical implications, her clarity of exposition?
I think that's like the 8th or 9th thing, as I kept getting more and more drawn to her as a central character, is when I saw her at the various conferences and the international summits and sort of reaching back to David Baltimore and Paul Berg, who had done the Asilomar process with the original recombinant DNA ethical issues and safety issues. And I even went with her, as you know from the end of the book, to go visit Jim Watson up in exile, where he's been in the house at Cold Spring Harbor. And I think that I've watched her or seen the videos of her talking to Senate group, the Senate and congressional hearings. So I think she explains in a genuine way the ethical issues. And to me, that was deeply important, as she says in her own book and expands in mine. She had that dream, that nightmare, that somebody wanted to learn about her technology. She walks in the room and it's Hitler. And that pushes her to engage. Yeah.
Let's bring in Alex into the conversation. Alex, have at it.
Sounds good. Hi, Walter. One point you're talking before about the rivalries, and in a sense there was one, but maybe not as much anymore. Over the past few years, you'd mentioned there'd been a seeming slight coolness between Jennifer Doudna and Emmanuel Charpentier, perhaps some resentment that Doudna was commanding public attention in the media by writing a book, but yet you help facilitate a conversation between them that may have helped heal some old wounds.
Yeah, well, as an old-fashioned journalist, I try not to insert myself in the story, but I liked both of them a lot. I spent time in, Berlin at the Max Planck Institute, and charmant. I speak French, but I never quite knew what charmant meant until I saw Emmanuel. I said, oh, that's what it means. And the fact there was no falling out, but there was a coolness, Alex, as you point out correctly. And, Emmanuel doesn't really love the limelight. Jennifer doesn't seek the limelight, but it aims at her a lot. And at one point, we were talking about the CRISPR conference at Cold Spring Harbor virtual this past fall was going to happen. And I said to Jennifer, because she asked me, because I'd just come back or I'd spoken to Emmanuel, what can she do? She really loves Emmanuel, Manu, she calls her. And, what can she do to warm things up again? I said, well, invite her to speak virtually at the CRISPR conference. And she did. And Emmanuel said no. And I said, well, Emmanuel, how about right after on Sunday when it ends? Why don't you and Jennifer just have a nice long talk? And they had this great talk. I mean, about kids, about everything else and stuff. So all of us have that. I mean, I could, I tried not to overhype rivalries or over extend these things. Because I think back at when I was editor of Time, the person who was editor of Newsweek, your friends, your rivals, your whatever, but then you get together. I also had a much more poignant experience in a different book, which you can go to the end of my Steve Jobs book. And Steve Jobs referred to Bill Gates in words that you cannot use on a family Zoom meeting all the time. And he was dying, Steve was. And they had had this incredible rivalry as well as cooperation. Anyway, at one point, Bill called me and said, what's the story? I said, it's pretty bad. I don't think he's going to beat it, in the next few months. He said, well, can I come down? I said, well, and Steve didn't do anything, didn't answer. So Bill flew down from Seattle and just went and knocked on the door of Steve Jobs' house. It's a scene in my book. And I tried to, I won't say facilitate it, but just I wanted this great ending where they both hug each other and get tears in their eyes. And they did. Bill said something like, your end-to-end control system of the operating system and the hardware, that ended up working better than I thought. And Steve said, oh, your license it anywhere system. And I thought, oh, that's a great ending. And then after they left, I said to Steve, that was nice. I'm glad you said that. And he said, he's such an asshole. And then just erupted. So I was not truly able to facilitate that. But I do think in the end, we're all part of a noble mission, especially scientists. And I think COVID helped us realize that. So I do hope that someday Jennifer Doudna and Fong Zhang and George Church all have coffee in Kendall Square, and they all do what Bob Noyce of Intel did with Jack Kilby, of Texas Instruments, when they were in a long patent battle and finally said, let's quit fighting over divvying up the proceeds until we finish robbing the stagecoach. And they just shook hands and crossed license. So maybe that'll happen.
That Steve Jobs book, by the way, a masterful work. Read that years ago. So,
well, thank you. I don't pretend that it was me. And with all due respect to Jennifer, who's a great central character, nobody will ever match Steve Jobs as being a colorful central character.
Mm. Now, you touched on COVID-19 a couple of times, most recently a few seconds ago. As you were reaching the closing stages of writing the book, the pandemic took place and the whole world changed, and this newsroom emptied out. The final 80 pages or so of your book turned to the scientific community's response to COVID-19. And what's your take from that twist in the world, let alone your story and story in the book, and how the scientific community responded, and what from that is likely to endure past the pandemic. For example, lightning fast reviews and approvals.
Yeah, well, certainly on journals, I think a little bit more open sharing and faster reviews, especially on things that are timely. But scientific journals have their really good peer-reviewed purpose and should not fundamentally change, in my opinion. I think there are a few things that will change. First of all, I hope, and I hope my book's a small part of it, that public interest in life sciences will be kicked up a whole quantum orbit. I mean, it'll be huge. Suddenly, how our molecules work is not only a subject of curiosity, but it's very useful to know. And along those lines, I'll pick one thing, which Feng was the one who kind of first planted in my head. But both him with his company, Sherlock Biosciences, and Jennifer with Mammoth Biosciences have been turning CRISPR into a detection tool for COVID as well as other coronaviruses. That was one of the astonishing failures of American society, of both technology and policy and science, it was just so hard to get a test. I mean, it shouldn't have been that hard. And now we've finally gotten these new detection technologies that don't use PCR, don't necessarily need to go through all the thermocycling, and so can be faster. And both Feng Zhang has showed me, and Lucas Harrington and Janice Chen at Mammoth have shown me these home testing devices with little cartridges, put a bit of saliva into it, and using CRISPR-derived technology, it can directly, quickly, and with great accuracy detect any genetic sequences. I think, and Kevin can correct me if I'm wrong, I haven't followed it much in the past month or two, they've gotten emergency authorization use for lab uses of this, but not to sell it at my Walgreens on the corner to take home. Everybody's nodding, so I didn't screw that up.
That's right.
But I think by the end of the year, we might, Kevin, go ahead, sorry.
No, I was going to say, I think that's right. And if not for this pandemic, maybe for the next one, this will really come into its own.
I think by the end of this year, some of it, at least they'll start rolling out. It's not just for the next pandemic. When Steve Jobs, pulled off this, which was, one of the great inventions of our time, he made it a platform upon which others could build. And so Ubers and Airbnbs and, Yelps and you name it, they all build on this platform. When the personal computer came home, I remember it very early on. I was an early adopter. I had a compact desk Pro. There is nobody on this Zoom screen that's old enough to remember the compact desk Pro. But it was, and then I had the, Macintosh, of course. When you got a home computer, a personal computer, it brought digital revolution into the home. It brought microchips into the home. You suddenly had a sense that you were connected to this digital revolution. When you get these home testing devices, whether it's the end of this year or beginning of next, and people are building things on it that say, oh, by the way, this cartridge will help detect a tumor if you have a sequenced tumor that you're trying to detect, whether it's still, whether it's, the genetic material is still around, you can do it this way. Or your gut microbiome, you might be eating the wrong yogurt. Or if kid has strep throat, or if you have the new coronavirus. And that technology will help connect the normal person to biology and biotech the way personal computers did to infotech. And that's going to be a lasting change from this pandemic.
Thanks, Walter. I think Juliana's got some audience questions. I'm going to lead off with one that came in before we started broadcasting, but it plays off the last question. What are the most interesting companies, Walter, working on CRISPR, and who, in your opinion, and who should we be paying attention to, whether public or private?
Yeah, I'm not great at that. In fact, so I'm not going to go too long on it. I do think Mammoth and Sherlock are wonderful, and so is Caribou with there's a chapter on the book. Rachel Horowitz starts coming out of Jennifer's lab for therapeutic uses. Certainly, Emmanuel has started CRISPR Therapeutics with Roger Novak, I think, and others. And that was the lead company, along with Vertex, that did the sickle cell trial that worked with Victoria Gray down in Nashville. And then, of course, Editas. And in my book, I don't know all the financial prospects for Editas, but I do know that really difficult square dance that happened with people going in and out and do-si-doing and Jennifer quitting that happened when the patent battle began.
Yeah, there's some lovely pages and vignettes on the musical chairs as the founders circle around each other trying to figure out, who was the dream team and who was going to go out on their own. It's great stuff. Juliana, do you have some audience questions for Walter, please?
Yeah, we have some great questions coming in. I'll ask a question from David, and he asks, how important was it for you to make a theme of women in science? And you're drawing the thread from Rosalind Franklin to McClintock to Jennifer Doudna.
Yeah, that's a really good question, because I sometimes ask myself this. I did not begin by saying, I'm going to write about women in science. And I was writing about this whole process. But yes, 6 years ago, 5 years ago, when I first started talking to Jennifer, and she told me the story of spotting Rosalind Franklin in the book, The Double Helix, and saying, oh my goodness, women can become scientists. And she told me about, meeting Barbara McClintock on the paths of Cold Spring Harbor, and how the women, in particular, Gillian Banfield, and then obviously Emanuela, pushed more on the RNA front. I realized that was an important strand of the book. So I did not, and Jennifer urged me not to overplay it. She said, I don't think of myself as a woman in science. I think of myself as a scientist, and I'm a woman, and I'm proud of my gender. I do think of a few things where women do face strong headwinds that are in my book, but not that blatant. Like when she and Rachel Horowitz were trying to start Caribou. It's a truly sexist community, the venture capital community. And they were like, fine, this may sound good. We'll find some guy to run it for you type. And they wouldn't invest. And so finally, Jennifer and Rachel and their families all invest to get Caribou started. So yes, women in science is a theme. It did remind me when she saw Ada Lovelace in that book and she decided to become a scientist. That's a really big deal. That was really good that her dad left that on her bed. Otherwise, she would have been, she wanted to study French. That was what she was planning to do. So I thought, wow, role models matter. So that's an added benefit of having Jennifer and Emmanuel and Gillian Banfield and others, especially Jennifer, on the cover of the book, is maybe somebody else will leave it on the bed of their daughter or their niece or their son, and maybe it'll be a role model.
Terrific. Thanks for that. Another question from an audience member, TJ. He says, I'm excited to read the book. You mentioned the scientific method, and he so appreciates you joining the method and going to conferences and the bench. Did that help you appreciate how scientists have to enjoy the victories to such a degree that drives through all the work and unsuccessful experiments?
Yeah, unsuccessful experiments are the heart of the scientific method. And this is not directly answering the question, but one thing that struck me when I noticed that is people, especially down here in Louisiana, they're ragging on the CDC, which probably deserves some criticism, but telling you not to wear a mask and then telling you to wear a mask and then doing things. Yeah, the scientific method works that way. You have a theory and then you get more evidence and people change their minds. And sometimes your experiments or the way you approach things turn out to be wrong. And so, there was a lot in my book where people thought CRISPR worked through RNA interference and marched all the way down. So did Gillian Banfield, marched all the way down that path. And many other things that they got wrong until they got it right.
From the audience, also, Akanksha asks, and it did sort of tie in with a question I had on issues of bioethics concerning CRISPR. How much did Jennifer Doudna and Emmanuel Charpentier grapple with the ethical implications of CRISPR gene editing, especially after He Jiankui's disclosures back in 2018 and other CRISPR researchers sort of weighing in, going another
Well,
direction?
both Kevin's book, Editing Humanity, and my book, and a couple of other books. There's a great Hank Greeley book that's just out that I really enjoyed called CRISPR People. Is that right, Kevin? Wrestles with it. I would say a quarter of my book, begins with Jennifer's Hitler dream, goes on to the conferences she starts to do, and there's quite a few chapters on the whole notion of there's Jennifer with He Jiankui and Michael Dean, the guy at Rice, who I think no longer at Rice, who was an advisor to He Jiankui. And so, I don't know, that comes 2/3 of the way through the book. So there's a lot of going step by step because there's no easy answers here. You got to go case by case. Like clearly we love what happened with sickle cell this past year. But I have somebody in the book that I went back to, David Sanchez, who's in this wonderful documentary, Human Nature, 17-year-old, loves playing basketball, except for when he doubles over in pain from sickle cell. And he's like one of the best bioethicists in the book because they say, we can edit your children so they won't have sickle cell. And he says, that would be wonderful. And then he pauses and says, but maybe we should leave it to the kid. And they say, well, would you want your kid to have sickle cell? He says, no, it was painful. I would not want that. But sickle cell taught me a lot. It taught me patience and perseverance, and it taught me empathy.
Yeah.
And I'm going, wow. And then I went back to him because I'm like, nobody can be that good. And so I don't really say, here are the answers to the ethical questions. But I think as with Kevin and as with Hank in their books, we kind of try to say, here are the people grappling with it, and here's what they're saying. And this is something we're going to have to decide. And by we, I don't just mean the scientists. I mean people like us who are science writers and people who are science readers and people who aren't science readers, but they should have some clue about this because society as a whole has got to make these ethical determinations. I think, and I write a chapter in the book about this, leaving it to pure individual choice could lead to a free market eugenics that could be as scary, almost not quite, as state-sponsored eugenics.
And your book, Walter, is sprinkled with anecdotes of even before the CRISPR babies saga, people writing to Jennifer and her team with some kind of pretty far-fetched, crazy ideas about setting up clinics, setting up companies. And even right after the CRISPR babies saga, we know that clinics are trying to set up offshore or start teaching courses on.
I have Sam Sternberg, Sam, as you know, Sternberg, and Healthy Babies Inc., where he's like recruited by somebody to do this Healthy Babies using CRISPR. But let me say one other thing. Also, the flip side happens. It happened yesterday during an LA Talks Live where Doris Kearns Goodwin was asking, isn't this bad? Isn't this editing going to be, shouldn't we shy away from doing even gene edits and living patients and stuff? And the chat room and the Q&A, and Jennifer told me it happens to her all the time, she starts filling up with people saying, I don't want to say this, alive, but I have a 12-year-old granddaughter and she's got, and they'll name the syndrome, and they say she's going to die in 3 years and her muscles are disappearing. And then somebody else will say, I've got this son and he's got this neurological genetic disease. Can you put me in touch with Jennifer Doudna because she might be able to save him? So before we get into all these people worrying about misuses of CRISPR, let's not get so frightened that we don't use it when there's enormous suffering caused by genetic problems.
Yeah, yeah. Juliana, do you have maybe a couple of final questions from the audience?
Sure, yeah. William asks, what have you found to be the most effective ways to raise interest in science in the general public? And is it too much to hope that we can raise some excitement about the future of science in both citizens and politicians?
Yeah, I mean, the way Kevin and I do it is edit journals, or whether it be Time or CRISPR journal, and write books. And as Kevin jokingly said, I have enough of a platform that if I write a book, I'm going to be on Andrea Mitchell, or I'm going to be on Morning Joe, or God knows what, Fareed Zakaria. And so we each should use our platforms that we have to say a few things, which is nature is beautiful. Basic curiosity should be the driving force of science because it leads to unexpected things. Basic curiosity research should be funded by the government because who knows when a coronavirus with a spike protein is going to hit us. And that science is a noble endeavor. I mean, I hope by raising awareness of science, I'll get myself again in trouble here, but some of my Tulane students, they tell me, okay, I'm really great in math and I'm going to join this hedge fund and I'm going to do this algorithm and, stuff. Well, maybe we can reallocate some IQ points from financial engineering to life sciences engineering, and that might actually benefit the planet a bit more if we do it wisely.
You have a chapter in the book in which you don a lab coat and go to Berkeley, and you're actually holding a pipette. I don't know if that was the first time, but you're running CRISPR experiments. Thankfully, they were supervised, and you even note that we washed the results down the drain to make sure there was no biohazard left in the lab. But what was that like?
It was Gavin Knott, who and Jenny Hamilton. I think both the graduate students. And yeah, I'm sitting there with the pipettes, and I did it in a historical way, meaning first we did the experiment that Martin Yinnick, Jennifer Doudna, Strelinsky, and Emmanuel Charpentier did, which is in bacteria in a test tube make an edit. Okay, that's great. And then, as you know, one of the great controversies that's still in the patent courts is how easy and how obvious it was to go from that to edit a human cell. So we had human kidney cells. I'm sure that's probably what people with training wheels like me get to use. And we made an edit in a human cell. And I was somewhat surprised how easy it was. I know creating the reagents is hard, but they said you could just order them online and stuff. So delivery mechanisms are hard, but doing the edit aren't that hard. And there's when Einstein went to school, and it was also the model of the school I went to, so Latin model, discimus agere agendo, which is simply we learn to do by doing. And I've always felt I'm not going to make an atom bomb, having written about Einstein, but I am going to try to take apart an Apple II computer and figure it out. And I am going to try to get into the lab and, even don a white lab coat, which will be my Mardi Gras costume next year when we have Mardi Gras.
Your book is almost like a coffee table book. You've shown us lots of wonderful photographs throughout the book. And what you also went out of your way to profile and heap praise on a new generation of CRISPR scientists. This isn't just about the big names, the Landers and Doudnas and George Churches, but another younger next generation. Were any people particularly helpful to you, or you think future biographers should be keeping an eye on?
Well, yeah, as I said, David Liu is somebody, and you don't have to be young to deserve a big biography. George Church deserves one, and Feng Zhang, who is still young in my mind, deserves one.
Yeah.
And yeah, it was kind of cool because people like Enrique Lin Xiao and Jenny Hamilton, who set up the COVID testing lab, their picture is in color in the book because they're heroes of the book. And Fyodor Yurnov and all of these people, and I don't think they're used to, and so they're like tweeting out, here's the chapter and look, So, but I do think, because I remember being at the West Coast Computer Fair and Comdex in the 80s and 90s, when techies and they're, creating things like Google were the stars and super celebrities. I think we should make, and George Church wrote about this in his response to Heroes of CRISPR, the Eric Lander piece. He said, we ought to make the heroes the people who are actually at the bench doing the experiment. So, Martin Yannick and Chris Strelinsky and Enrique and others. And as for the book, we both sort of share a publisher, Kevin, meaning I think we're both in the Simon & Schuster family. Is that right?
Pegasus is distributed by Simon & Schuster.
Yeah. One of the things I've earned, besides a bit of a platform that allows me to get on Andrea Mitchell or something, is I can go to Simon & Schuster and I can say, I want color pictures and I want high quality paper. And I want the color pictures throughout. I don't want them just in insert. And I don't want you to charge any more for the book than, every other nonfiction book like that. And I think because having done that with Leonardo, where you truly needed it, and with Steve Jobs having, I had some great pictures there, it does help both popularize it. People can just open it up and say, wow, there she is in her kitchen winning the Nobel Prize. But just a little tip to you that if you push your publisher hard enough, they're going to give you color pictures. It's not that hard.
I got a bunch of color pictures, but they're all stuck in one sleeve in the middle. But that's fine. I was thrilled.
They told me that they had to put it in that sleeve in the middle because then they couldn't. I said, no, you don't. I mean, fortunately, I've worked at a magazine for many years, and I said, no, you don't. They can run color throughout.
Well, we're no Walter Isaacson. What is maybe you can break some news for us. What's going to be the subject of your next book?
I really don't know. I sometimes go in the Wayback Machine, as I did with obviously Leonardo. And after writing about, sometimes like a Steve Jobs, it's like, all right, let me go do somebody in the distant past. I loved working side by side with people. And so I probably will do another current person. I'm very interested in Bill Gates because he's, like Jennifer, every field of endeavor almost, from small nuclear power plants to the original BASIC for personal computers. He's interesting to me. Elon Musk, of course, is a Steve Jobs-like character, and I'm totally fascinated by it. I think he's brilliant and he's changing things. I fear that at least right now we might be in the middle of the movie, so to speak, and I want to see how the movie unfolds. I wrote a mini biography of Jeff Bezos. That's the introduction of his book that came out a few months ago called Invent and Wander. But he too is sort of in the middle of many transitions, let's say. So if you have other ideas, I've also been interested in Marie Curie, of course, because physics and chemistry. I don't know, Kevin, give me some ideas. You're an editor.
Let's take that offline because I think you've come up with half a dozen that are better than anything I would come up with. John, Alex, Juliana, time for one last question. Any burning questions that you'd like to throw in there?
I'll be fast.
John, go ahead.
Yeah, just very quickly. So in the beginning of your book, you talk about at the beginning of this terrible pandemic, Doudna got all these people together with Zoom and said, we need to step up. What does she mean by that? And how did she and her colleagues do so?
I think very specifically, she meant that the testing fiasco was already apparent by exactly a year ago, I think this is early March scene in the book. And it was staggering to me, but more so to her, that people who want to be tested for COVID, the firefighters in Berkeley, the teachers, the professors, they couldn't get tests. And we had a president, and I don't want to get political, he kept saying, everybody get tested. Anybody wants a test, have a test. And me, I'd go down to the clinic, I couldn't get a test, So initially, it's like we're going to take every biology lab in this country, from Berkeley to Stanford to the Broad, whatever, and we're going to set up testing facilities, which is what they did. And then she said, we're going to divide into 10 teams. And the 10 teams are in there, but one of them is, let's not use PCR. Let's just use a quicker direct test we can do at home. Likewise, can we turn CRISPR into an antiviral? which still hasn't been done yet, but Pac-Man, which is the name of Stanley Cheese in Jennifer's orbit. CRISPR journal has written about it. And then I think Carver, which Cameron Mirvold has done, I sort of say in the Feng Zhang orbit, meaning he comes out of Cambridge. So they broke themselves into teams like that. And I think they were very disappointed that the policy and government were screwing up, especially testing and detection, but also data collection, data analysis, and finding antivirals. She did not throw herself into vaccines. She knew quite well the power of messenger RNA, as well as almost anybody in the world did. And she knew that that was progressing quite well.
Rodolphe just texted me to say CRISPR is, of course, the ultimate antiviral. So it'll be very interesting to see how that field pans out. That's all the time, unfortunately, that we have time for today. We could carry on chatting with Walter all afternoon, but he's got to prepare for a Tulane lecture. So we need to let him get some lunch and go off and do that. Thank you, Walter. It's been an honor to host you. Thank you, everyone, for watching. Thanks to Rodolphe Barangu and the CRISPR Journal for sponsoring today's episode. And join us on GenLive. We do this every month, not always with best-selling authors, but next month we'll be turning our attention to synthetic biology, and I think that'll be a lot of fun as well. GenLive is produced by Jamie Cohen and Liana Jabs, and for the entire GenLive team, thank you for watching. I'm Kevin Davis. Stay safe, and goodbye for now.
Thank you. Thank you.
Thanks, Walter.
Walter.
Thank you, John.